The effect of light intensity on the rate of photosynthesis by Katie Pratt's group.
Oops.
She's not going to know who's...
Okay, in our abstract, we said this experiment was performed in order to determine the effect of light intensity on the process of photosynthesis. We tested different intensities of artificial light on a simulated situation of photosynthesis. Dark, no direct light, intense, and normal light intensities were tested. After conducting our experiment, in which we measured the ET50, or the time it took half of the leaves to float, of deoxygenated spinach discs to float in a bicarbonate solution, it was determined that the normal intensity of light was the most effective in the rate of photosynthesis, disproving our original hypothesis that the more intense light would be the most effective on the rate of photosynthesis.
The purpose of our experiment was to determine the relationship between intensity of light and photosynthesis. We initially hypothesized that the higher intensity of light would result in an increase in the rate of photosynthesis, which we measured by the ET50, because we assumed that since the light-dependent reactions of photosynthesis are what produce oxygen, more light could only mean more oxygen production. The production of more oxygen would theoretically cause the disk to rise faster, lowering the ET50, which would in turn suggest an increase in the rate of photosynthesis.
Materials, methods, procedure. Materials: syringe, spinach leaves, hole puncher, cup, dropper, dish soap, light lamp, stopwatch, water. Procedure: first step, punch holes in the spinach to get discs. Second step, drop 1 to 2 drops of soap into water in the cup. Third step, place spinach into syringes. Fourth step, take in 5 milliliters of solution into syringe. Fifth step, vacuum syringes so that the leaves sink in the solution. If they do not sink within 3 tries, restart. Sixth step, immediately put leaves in cup with solution. Prior to this, adjust light lamp to properly measure that is being tested for that trial. Seventh step, start stopwatch and time how long it takes for the spinach leaves to rise or until it is deemed that they will not rise. Eighth step, repeat for each level of light.
So the results of our experiment showed that the normal light intensity, which was 12 centimeters from the surface of the solution, was the fastest with an ET50 of 4 minutes. The intense light, which was a half centimeter from the surface of the solution, had an ET50 of 5 minutes, and both the dark and the no direct light test had no photosynthesis occur. As you can see here, there was an inconsistency with... It's recording that. Okay. There was an inconsistency between all the experiments as the normal light and the no direct light. The normal light only took 6 minutes for all the leaves to float, where the no direct light, we stopped collecting data because none of them were floating. And then with the intense light, which we predicted would be the fastest, but it wasn't, at 7 minutes and until 10 minutes, the last leaf stopped floating, so we presumed that one might be dead.
Basically, the conclusion we reached was that light has a profound effect on photosynthesis, clearly, and that is evident by the graph here. And out of control in the dark, no photosynthesis occurred, none of the leaves floated. The interesting thing was our result. At a higher light intensity, which is about 1 centimeter to the surface of water, showed a lower rate of photosynthesis than at a more moderate intensity, 12 centimeters away from the top of the water. It is important to acknowledge the fact that we cannot conclude if this trend continued further out, for we only performed 3 trials.
The end.
El fin
I did the introduction, by the way. That was
de
me.
semana.
Introduction.